The transfer of genetic information between organisms in the same generation is called horizontal gene transfer. 3 types of horizontal gene transfer occur in bacteria: transformation, transduction, and conjugation. All 3 mechanisms involve a donor cell that transfers a portion of its DNA to a recipient cell, which may incorporate the new DNA into its own, becoming a recombinant organism. During conjugation in E. coli, a plasmid is transferred from the F-positive cell to an F-negative cell. As a result of conjugation, the F-negative cell receives the F vector, becoming an F-positive cell. The unencapsulated cells of the rough or R strain of Streptococcus pneumoniae do not cause disease. When mice are injected with these cells, their immune system efficiently deals with this invader and the mice live. Cells of the smooth or S strain of Streptococcus pneumoniae produce a capsule that allows them to escape phagocytosis by the body's defensive white blood cells. When mice are injected with these cells, the mice die. Heat can be used to kill the smooth strain of Streptococcus pneumoniae. When mice are injected with these heat-killed cells, the mice live. Surprisingly, when mice are injected with a mixture of harmless, heat-killed, smooth-strain cells and harmless, live, rough-strain cells of Streptococcus pneumoniae, the mice die. Live, smooth-strain cells can be found in the blood of these dead mice. The harmless, rough-strain cells must have gained the genes to produce capsules, like those of the virulent, smooth-strain cells. Some bacteria are capable of taking up fragments of DNA from their surroundings and integrating the fragments into their own chromosomes by recombination. Cells capable of taking up DNA from their environment are said to be competent. Only a few genera of bacteria are naturally competent, such as the S. pneumoniae in this example. But other bacteria, including E. coli, can be made competent through simple laboratory treatments. In transduction, DNA from the chromosome of one cell is transferred to another cell via a replicating virus. A virus that infects bacteria is called a bacteriophage, or phage. At the beginning of generalized transduction in bacteria, a bacteriophage attaches to a bacterial cell wall and inserts its DNA. The phage commandeers the bacterial cellular machinery to synthesize new phage DNA and produce phage proteins. In order to completely convert the bacterium into an efficient virus-producing factory, some of the phage proteins break the bacterial DNA up into small pieces. Normally, phage DNA is coated with phage protein to form new virus particles, but sometimes pieces of bacterial DNA are mistakenly packaged in phage proteins. These sloppily packaged phage particles carry bacterial DNA rather than phage DNA and are called transducing phages. When a transducing phage, composed of phage proteins and bacterial DNA, infects a new host, the phage injects the bacterial DNA into the new bacterial host. The infected bacteria sometimes incorporates this DNA into its own chromosome through recombination, resulting in the transfer of genes from the donor bacterium to the recipient bacterium.